Okay, so my name's Emma Hodson-Tole. I'm a reader in neuromusculoskeletal integration in the School of Healthcare Sciences at Manchester Metropolitan University. My research is very much interested in skeletal muscles, so the muscles that enable us to move, and that research focuses on the muscle structure and function and the integration between those things. So how does the size and the shape of a muscle link to the roles that it enables us to complete. I used a technique called electromyography, which enables us to record the electrical activity that muscles generate as part of being activated. So when a muscle turns on to contribute to a movement or produce force to enable a movement to happen, there's electrical activity occurs, and we can record those electrical signals and then do some different mathematical processing with the signals to give us different information about how hard the muscle was working, how long it was working for, and different aspects about some of the properties of the muscle fibres which contributed to the particular movement that we looked at. One of the main research projects that I'm currently involved with is a project which relates to the diagnosis of neurodegenerative diseases like motor neurone disease, which is also known as ALS, so MND or ALS. And what we're interested in doing within that project is helping the clinicians by potentially providing another tool for diagnosis. So at the moment, the diagnosis of the disease is based on recording electrical activity in the muscles using electromyography, but with needle-based electrodes, so it involves inserting needles into the muscles of interest. And what the clinicians are looking for are involuntary twitches that the muscles undergo, which are an indicator of the fact that some degeneration is taking place, some changes are happening in the nervous system. Now, the electrodes which are used only record from a very small part of the muscle, which means the clinicians are not able to get a big picture, and there might be indicators which are missed. What we want to use to help the clinicians with the diagnostic process is a technique called ultrasound imaging. Now, ultrasound imaging is used in hospitals quite commonly for things like scanning unborn babies. So it's a very safe technology that's non-invasive. What we can do is place the ultrasound probe over the muscles we're interested in scanning and get images of how those muscles are behaving. So are they just statically still, or are we seeing local changes and local movements in the muscle? So our research is trying to collect images from people who are affected by motor neurone disease and looking at how the involuntary twitches appear in collected images, and then develop computer analysis techniques to automatically analyse images in an objective manner. The research that I'm involved with potentially gives clinicians a new, more sensitive way of diagnosing the disease, and critically it potentially means we can identify the disease earlier in the process, which means that the drugs that are developed, or hopefully will be developed in the future, can be administered at a stage when the nervous system is relatively healthy, and therefore the drugs have a better chance of being more effective. The research also gives us the potential of having a monitoring tool, so we can very sensitively detect changes in the system, either deterioration or hopefully in the future improvement as the result of new drugs which will be developed as a result of other research lines which are occurring at the moment.